Okay, let's talk about ivermectin. Ivermectin is like hydroxychloroquine in that it's an FDA-approved medication that already has a purpose for some other infection, but it's looking like it might be helpful with COVID-19. Ivermectin is usually used for parasites, and it has been investigated in other viruses like West Nile virus and influenza. But now researchers are looking at this for COVID-19. To get a better understanding of how this ivermectin actually works, we've got to go back to cellular biology. So this is the cell, of course. Here we have the nucleus. We've got DNA here in the nucleus, and we've got a protein here in the nuclear membrane that allows the messenger RNA to come out into the cytoplasm and allows it to be translated into protein. Well, the problem is that the virus, when it comes in, it also has its version of RNA. And of course, the ribosomes can't tell the difference. And so they start to translate the messenger RNA from the virus into viral proteins. And these viral proteins can have problems. They can have unintended consequences. And these proteins can come in and go through these proteins that allow the viral proteins to go into the nucleus where they can cause problems. And that could lead to decreased immunity, especially against this virus. And some scientists believe that this is actually what's going on with the coronavirus. There's a specific target for this transporting in or importing in. And cleverly, they call this protein the important protein. And there's several different versions of this. There's an important alpha, and there's also an important beta 1. So otherwise, this is known as imp alpha and imp beta 1. And what ivermectin does is ivermectin shuts down this ability to transport in proteins, viral proteins in this case, into the nucleus, which diminishes the ability of the virus to cause harm to the cell. So this article that was published on the 3rd of April, 2020 goes into that data. And the version that I'm showing you here has a link in the description below, and it is a journal pre-proof of publication. And the summary of the article is, although several clinical trials are now underway to test possible therapies, the worldwide response to the COVID-19 outbreak has been largely limited to monitoring and containment. We report here that ivermectin, an FDA-approved antiparasitic previously shown to have broad-spectrum antiviral activity in vitro, is an inhibitor of the causative virus SARS-CoV-2 with a single addition to the Vero H SLAM cells. Those are the type of cells that they use here in the test tube. 2 hours post-infection with SARS-CoV-2, able to affect a 5,000-fold reduction in viral RNA at 48 hours. That's a pretty short amount of time and a large amount. Ivermectin therefore warrants further investigation for possible benefits in humans. And this, of course, is weighty because this medication is already being used in humans and has been deemed to be safe enough to be used in humans. So after giving the cells ivermectin, they looked at day 0, 1, 2, and 3 to see what the relative viral load of RNA was in terms of percent. And you can see here over a couple of days, it drops 1, 2, 3, 4, 5 orders of magnitude in just a 48-hour period. So that's inside the cells, the amount of virus inside the cells. Supernatant is referring to the solution outside the cells. And here you can see over a couple of days, we have a 1, 2, 3, 4, almost 5 orders of magnitude drop in the amount of relative viral RNA. Here, they're looking at the RNA-dependent RNA polymerase gene, which we talked about a number of weeks ago, is the gene that's coded for by the RNA in the virus, and that is also known as replicase, which causes gene replication. Here we can see that ivermectin at specific concentrations completely knocks out that genome. And here we see the medication ivermectin inhibiting this IMP-A and IMP-beta-1 from allowing the coronavirus proteins from going into the nucleus. You see, when they go into the nucleus, the hypothesis is that there's an enhanced infection and reduced antiviral response. When ivermectin binds IMP-alpha and IMP-beta-1, it prevents it from binding to the coronavirus protein and importation through that nuclear membrane so that you have infection not enhanced, and there is a robust antiviral response that occurs. So this research is interesting because it does address the issue regarding immunity that we've discussed over the last couple of weeks. And it also correlates with what I've personally seen in the hospital, with patients arriving in the emergency room only to decompensate within the next 12 to 24 hours and die. Which tells me for some of these cases that the only way you're going to be able to intervene is in those precious hours and days before decompensation. This mechanism that we're seeing here of reducing the immunity, reducing the ability to mount an immune response is an important one.